Hello friends, this is Dr. Pankaj Kumar, your mentor, and I welcome all of you once again in another session of the Need Why. In today's sessions, we will be talking about bacterial cell wall. Now, the moment we talk about the eubacteria cell wall, right? So you know very well it is a dead rigid structure which basically provides protection, and it is chemically made up of peptidoglycan, right? Now, peptidoglycan is actually made up of two things. One, amino sugar, and second is amino acids, right? And we know very well that there are two types of amino sugar, that is NAN and NAG. So NAG stands for N-acetyl glucosamine and NAM stands for N-acetyl muramic acid. Have a look at this. So you can see there. So this is the structure of the cell wall. So you can easily see that beta-1,4 glycosidic linkage is between NAG and MAG. So this is a NAG and NAM, as you can see there, and this is the beta-4 glycosidic linkage, what you can see over there. So glycosidic linkage is going to occur between carbohydrate. So this is the beta-1,4 glycosidic linkage that is going to occur between the two carbohydrate, that is NAG and NAM. Okay. So what we see that in this peptidoglycan structure, there are 4 amino acids that is present laterally linked with a side chain, right? And these 4 amino acids are very, very specific, and they are specific in terms of their presence in gram-positive and gram-negative bacteria. For instance, you see the 4 amino acids are L-alanine, D-glutamic acid, D-alanine, and L-lysine. These are found in gram-positive, right? And rest of the 3 is going to occur in gram-negative also, but in addition to, in place of L-lysine, what we see, there is a diaminopimelic acid that is present in case of gram-negative. Okay, so this question is often asked in competitive examinations that what are the or which type of amino acids is actually present in gram-positive and gram-negative. So once again, I would like to reiterate that L-lysine exclusively found in gram-positive and diaminopimelic acid exclusively found in gram-negative bacteria. Right? So you see, another very important aspects in the bacterial cell wall is the stain. It was a stain, the most popular stain, what we call gram stain, right? Now, what is gram stain? So gram stain consists of crystal violet. So basically, it helps in identification of the nature of the cell wall of the bacteria. Okay. So all those bacteria who retain the gram stain, we call them gram positive, and the bacteria who fail to retain the gram stain, we call them gram negative, right? One more. In gram positive, we see there is a teichoic acid that is exclusively present in case of gram positive bacteria, right? So we will see the various steps of the gram staining. Okay, so what are the steps? So altogether, we can divide gram staining in 4 steps. The very first step is that bacteria are stained with the crystal violet. Naturally, the bacteria become violet. Then iodine is added. Iodine is going to act as a moderant, and they will form a crystal violet iodine complex. Then it is washed by alcohol, preferably the ethyl alcohol, right? So they decolorize, and the counter staining is actually done by the safranin. Okay. So have a look at all these 4 steps in these diagram. Okay, first you see I have shown gram positive and gram negative bacteria. So ideally they look alike. So when I put a crystal violet, both are going to look as a violet, right? And when I wash by iodine treatment, they will appear as a violet, as you can see there. And then we apply ethyl alcohol, then what we will see that gram positive is going to retain the crystal violet, but gram negative, you can see there that this is a white. They are not retaining the crystal violet. Okay. And when it is actually counter stained by safranin, you see that it is going to be appear as a pink, right? So gram negative appear pink and gram positive is going to appear as a violet. Okay. So this is all about the gram staining. Now there is a one big question that why gram stain is actually not retained by the gram negative. And for this, we need to understand the structural detail of gram positive and gram negative. Okay. So what happens in gram-negative bacteria, at the outer layer, there is a LPS, lipopolysaccharide, right? And lipopolysaccharide prevents the entry of gram stain to the peptidoglycan layer. Okay. So the outer membrane is when it is going to be permeabilized by acetone treatment, and when the pink stain is counter stain is actually put over there, right? Then the safranin reach to the peptidoglycan layer and it make it a pink. Okay, have a look at this diagram. Now this is the cell wall of gram negative, and I would like to draw your attention towards the relative positions of the peptidoglycan, the lipopolysaccharides, and the membrane. You see there, the outermost layer is the lipopolysaccharides, right? Then in the mid, there is going to have a peptidoglycan, and the inner side there is going to membrane. So what will happen? When we add the crystal violet, right? Initially, it becomes violet, but it fails to penetrate to reach at the peptidoglycan layer. So when alcohol is put over there, the entire thing actually get washed. But when we put acetone, what happens? Acetone is actually, or what we say, when we put a safranin that is actually prepared in acetone, then acetone actually what they permeabilize this particular layer and they reach the peptidoglycan layer. So the safranin binds to the peptidoglycan layer and they gives a pink color, right? So this is about the gram negative bacteria. On the contrary, if you see gram positive, in case of gram positive, what you see, the outer layer is peptidoglycan, inner layer is a membrane, right? So when we put a crystal violet directly stain the outer peptidoglycan. So I think it is very clear that why the gram stain is not retained by gram negative bacteria, but actually it is retained by the gram positive bacteria. Okay. Finally, let's see that how we can differentiate between gram positive and gram negative bacteria. So first things, 80% of the cell wall of gram positive bacteria is made up of peptidoglycan, whereas in case of gram negative is just 15 to 20%. Lipopolysaccharide is not present. Lipids, though it is present, but that is present in a very, very minute quantity. But lipopolysaccharide is present in case of gram negative bacteria, and lipid content is also high, that is 10 to 15%. Another thing, teichoic acid, right? Teichoic acid is going to present in case of gram positive, but teichoic acid is not present in case of gram negative. You know, what is the function of teichoic acid? Teichoic acid some sort of provide as a receptors, right? Later on, when we will discuss in biotechnology, then I will make you understand in a deeper manner the very role of the teichoic acid. But here what we will conclude that teichoic acid is present in gram positive, it is not present in case of gram negative. Already I told you, diaminopymalic acid, that is one of the amino acids that is present in gram negative, not in gram positive. But in case of gram positive, it is a L-lysine, right? So in gram negative, there is a diaminopymalic acid and L-lysine is absent. As far as gram positive bacteria is concerned, mostly they are coccus and uncapsulated, right? But gram negative bacteria, mostly bacillus, that is a rod shaped, and they are capsulated. Okay? And finally, gram positive bacteria produces exotoxin and gram negative bacteria produce endotoxin. You might be wondering, what is the difference between exotoxin and endotoxin? On a simple language, I can make you understand that the biting of honeybee is actually exotoxin, the biting of a snake is a endotoxin. And as far as the mode of action is concerned, the exotoxin is going to infect or affect your peripheral nervous system, but as far as endotoxin is concerned, they affect the CNS, that is central nervous system. So that is how we differentiate between gram positive and gram negative. Right? So that's all as far as the entire bacterial cell wall is concerned. This is the most important, and in NEET, the questions are often figured from the bacterial cell wall, mostly concerning the difference between the gram positive and gram negative. Do join me in the next session when I will be talking about the bacterial protoplast. Right? Thank you.